my name is Crystal Santos, and today I will be doing my Make a Splash presentation on why the Limulus polyphemus, commonly known as the Atlantic horseshoe crab, should be aquacultured. I will discuss what makes the species so valuable and how aquaculturing them will benefit the medical community, improve sustainability, and combat one of the greatest threats, which is overharvesting. Horseshoe crabs have been roaming this planet for over 450 million years and have survived 5 mass extinctions, making them older than dinosaurs. They can be found in the Atlantic Ocean along the North American coastline and along the East and Gulf Coast, as well as Mexico. Despite their name, horseshoe crabs are more closely related to scorpions and spiders than actual crabs. They have a lifespan of around 19 years and take up to 11 years to reach sexual maturity. Males are generally smaller than females at maturity. Horseshoe crabs can survive a wide range of environmental conditions, including variations in salinity, and usually found in the sand or mud flats, estuaries, and mangrove swamps. They lay their eggs on coastal beaches in late spring and summer. Each year, when the horseshoe crabs arrive on the beaches of the Atlantic coast to lay their eggs, 500,000 of them are rounded up and brought to the lab and bled alive to extract a substance in their blood cells called Limulus amoebocyte lysate, which I will refer to as LAL. Their blood contains a copper-based oxygen-transporting protein called hemocyanin. They drain about 30% of their blood volume. 30% of the crabs do not survive the process. The survivors are returned to the water, but no one really knows how well or if they recover. In 2016, the International Union for the Conservation of Nature bumped the American horseshoe crab up to vulnerable on its red list, one step below endangered. It had been projected that wild populations could keep declining by as much as 30% over the next 40 years. Their blood is so valuable that it is the basis for a multi-million-dollar pharmaceutical industry, and a single gallon costs $60,000, making it one of the most valuable liquids on Earth. Current global fishery production is severely threatened by overfishing, climate change, and their interactions. However, with the advance in aquaculture techniques, this opens up the possibility to release hatchery-reared juveniles to help restore depleted global fishery stocks. Stock enhancement through captive rearing of juveniles is an important alternative to repopulate the native population, which in turn supports sustainable resource utilization and research activities. There is a high demand in the medical industry. Horseshoe crab LAL is used in sterility testing, and it ensures drug and medical device safety for millions of patients each year. There is a competitive economic potential that could secure jobs for more scientists and researchers to work in the industry. Eliminate the pressures on wild species due to harvesting, as they have potential to practically harvest LAL from aquacultured horseshoe crabs. The harvest could be broken down into 2 distinct 10% extractions per month, or potentially 12 to 24 times annually. There are also global conservation benefits from species recovery in North America and preservation in Asia. In turn, an expanded global supply of sustainable LAL raw material would provide a compelling basis to develop new clinical agriculture, veterinary, and food safety applications. Researchers in this article designed a controlled aquaculture habitat for horseshoe crabs and evaluated the effects of captivity on health markers such as amoebocyte density, hemocyanin levels, and LAL activity. They used recirculating aquaculture system and equipped them with 2 holding tanks, a biofiltration tank, and a clarity tank. The stocking density of the tanks in this experiment was 1 animal per 3 square feet. A health assessment was recorded for each horseshoe crab upon intake and were sourced directly from the wild. They found horseshoe crabs aquaculture to be practical, with routine harvesting resulting in high LAL quality while safeguarding animal well-being with 100% survival. In the figure, you will see 2 images from the article that display the LAL gel clot test. The researchers in this article summarize the available captive breeding and rearing techniques discuss the release strategies that could potentially improve the survival of released juveniles, and identify the future opportunities of responsible stock enhancement programs. Most available rearing results were derived from laboratory experiments in which their practicability in large-scale seed stock production is virtually unknown. The mortality of released juveniles is typically higher than 60% within the first month. Potential reasons that can cause high mortality of released juveniles include predation, habitat condition, environmental stress, and extreme weather. To improve the survival of released juveniles, they should be placed on the intertidal areas during low tides in summer when the surface water temperature exceeds 20 degrees Celsius. This can also mimic the spawning period of horseshoe crabs, typically recorded in late spring, and would coincide with the summer peak in juvenile production. Proper management of stress derived from handling and transport can also improve the survival of released juveniles. Unfortunately, to the best of our knowledge, no known study has focused on the effects of handling and transport stress on horseshoe crabs before release. The figure displays the life cycle and the associated habitats for horseshoe crabs. Little is known about the practicability and effectiveness of releasing large numbers of hatchery-bred individuals through release programs. Challenges such as producing cost-effective juveniles, developing optimal release strategies, and searching for an appropriate monitoring and evaluation methods. Despite the recent progress in aquaculture, the mass production of environmentally fit juveniles for release has been largely impeded by the presence of undefinable captivity-related issues due to the knowledge gaps in understanding the growth requirements and complex life histories of the horseshoe crabs in their nursery habitats. In conclusion, horseshoe crabs are vital to human health, medical research, and safe vaccinations. Aquaculturing them could enhance the stock density, restore depleted populations, and eliminate the pressures of harvesting. I think it's a win-win situation for both humans and horseshoe crabs. Here are my references, and thank you for listening to my presentation.